This module will focus on what happens after the cells have proliferated, migrated, and aggregated. As stated earlier, in order for our nervous system to develop well into the structure that we need to have for normal behavior, we find that migration and aggregation are necessary. However, once completed, then there is a process of growth that occurs, and that's what we talk about projections. The projections we're talking about are the axons and the dendrites of each cell. Keep in mind that the cells that were migrating at this point were in immature form. That means they did not have axons and dendrites. It was more so the soma, the body, that was just migrating across. In order for the nervous system to function correctly, these projections must grow to the appropriate targets. They cannot be growing to areas where they are not needed, as they would bring about dysfunction. We understand that the growth seems to be consistent in a fashion that's guided by several chemicals. The growth cone is basically the growing tip of each projection, and you may be able to appreciate some of that right here. This is what extends and retracts the filopodia as if it's finding its way, similar to how we saw the cells migrating earlier. Per a study in 2009, it was discovered that unlike what Roger Sperry, an influential biopsychologist, discovered, instead of growth cones being influenced by the target location, they actually seem to be influenced by a series of chemical signals along the route. So it's not that the cone is actually attracted by an area over here, if you will. It would be that there are chemicals all throughout the pathway that are guiding it continuously to get there. Axonal growth is very important, as it is through those that we are able to carry the neural impulses across to other dendrites. In a classic study done by Roger Sperry in 1963, Sperry cut the optic nerves of frogs, rotated their eyeballs 180 degrees, and then waited for the axons of the retinal ganglion cells, which compose the optic nerve, to regenerate, that is, to grow again. Once regeneration was complete, Roger Sperry used a convenient behavioral test to assess the frog's visual capacities. And you could see that in this particular picture. Under normal conditions prior to the surgery, when the insect was dangled in front of the frog's tongue would actually strike accurately right in front of it. And that is because the projections, the axons, were connected to the specific location. However, when the rotation had been made, and you see how blue is now at a different place, the frog would actually misdirect its strikes by 180 degrees. And so you could see that while the insect is over here, the frog would be striking on this end. Please note that this is occurring without cutting the axons. However, when the optic nerve is cut, and you can see now that there's a disconnect in here, then initially the frog would be blind, but when the optic nerve has been regenerated, then the frog would misdirect its strikes in a similar fashion to the top one here. This is because the axons of the optic nerve, although rotated, grow back to their original synaptic sites. Consistent with his observations, Sperry proposed the chemoaffinity hypothesis of the development of our axons. He basically said that each postsynaptic surface in the nervous system releases a specific chemical label, and that each growing axon is attracted by the label to its postsynaptic target during both neural development and regeneration. Once again, please note that per studies done in 2009 by different researchers, it was discovered that a growing axon is not attracted to its target by a single specific attraction released by the target, as Sperry thought. Instead, we know that growth cones seem to be influenced by a series of chemical signals all along the route. From different studies, we've been able to conclude that there are growth cones that take a role of pioneers, and that is to say that they are the first to travel in a route that's never been traveled before, kind of mavericks in their own respect. And they do so by interacting with guidance molecules. And we know that this, again, is continuously provided throughout the entire route. We also know that there is fasciculation, and that is an arrangement in bundles, the tendency of developing axons to grow along the paths that have already been established by the pioneer growth cones. So once this one occurs, then there's a higher possibility that other axons will grow in bundles, and this would allow for a proliferation of cells within there that gives way to the complexity of our brain and the many different connections. And the third understanding of this in here is that there is a hypothesis that seeks to explain how it is that our maps are laid out in a topographic fashion. That is to say that our eyes seem to be consistent from the retina to the geniculate to the thalamus, and they do not seem to fluctuate or vary much. Same thing with ears, and even when we looked at pain, we find that cutting specific parts of the cerebral cortex that would be responsive to a specific part of our body does not eliminate pain. And this is because of the many different pathways through which we can rearrange. And so the topographic gradient hypothesis is what has been used to explain how the topographic maps exist in our brain. Once the axons have reached their intended sites, whether it be through pioneer growth cones or fasciculation, they must establish an appropriate pattern of synapses. A single neuron can grow an axon on its own, but it takes coordinated activity in at least two neurons to create a synapse between them. This is one reason why our understanding of how axons connect to their targets has lagged behind our understanding of how they reach them. Synaptogenesis, which is the formation of new synapses, requires high levels of cholesterol that is actually supplied by astrocytes. And this is why the presence of glial cells is so important in there. Please note that we often think of cholesterol as bad cholesterol, but cholesterol, there's also good forms of that, and this is a compound of steroid type that's found in most body tissues, including the blood and the nerves. And we find that cholesterol and its derivatives are important constituents of cell membranes and precursors to other steroid compounds. Early theories about the contribution of astrocytes to synaptogenesis emphasize the nutritional role. Developing neurons need high levels of cholesterol during synapse formation, and an extra cholesterol is supplied by astrocytes. One of the complications researchers face in understanding synaptogenesis is understanding the promiscuity that developing neurons display when it comes to synaptogenesis. That is to say that they get attracted to several different connections. Although the brain must be wired according to a specific plan in order to function. And in vitro studies suggest that any type of neuron will form synapses with any other type. However, once established, the synapses that do not function appropriately tend to be eliminated. And so this variety of signals that act on developing neurons is what really makes the understanding of synaptogenesis quite complicated. Everything that's good must come to an end. And so we find that this amazing proliferation and migration and aggregation and all these amazing processes through and the journey that our cells have gone through to develop our nervous system also requires a component of death. And so we'll be talking about how neuron death and synapse rearrangement are essential also for our functional brain. Neuron death during development was initially assumed to be a passive process. It is believed that we have about 50% more neurons than we actually need. And so death is normal and important for us to be able to function well. It was assumed that developing neurons died when they failed to get adequate nutrition. However, it is now clear that cell death during development is usually active. By active, we mean that there are genetic programs inside each neuron that seem to be triggered and eventually cause the cells to actively commit suicide. A passive cell death is what we would call necrosis, whereas active cell death is what we would call apoptosis. Apoptosis seems to be safer than necrosis. Necrotic cells break apart and spill their contents into extracellular fluid, and the consequences is potentially harmful inflammation. In contrast, in apoptotic cell death, the DNA and other internal structures are cleaved apart and packaged in membranes before the cell breaks apart. These membranes contain molecules that attract scavenger microglia. You may remember those are the ones that heal and engulf and process the whole regeneration process, and other molecules that prevent inflammation. Apoptosis removes excess neurons in a safe, neat, and orderly way, but apoptosis has a dark side as well. If genetic programs for apoptotic cell death are blocked, the consequence can be cancer. If the programs are inappropriately activated, the consequence could be neurodegenerative disease. If this doesn't support the notion that neuron death is important, we don't know what will. Please again keep in mind that the neuron death is essential for a healthy brain. Evidence that life-preserving chemicals are supplied to developing neurons by their postsynaptic target come from two kinds of observations. One, through which they grafted an extra target structure, like an extra limb, to an embryo before the period of synaptogenesis. And what this did is that it reduced the death of neurons growing into the area. And destroying some of the neurons growing into the area before the period of cell death increased the survival rate of the remaining neurons. So you can see, again, here how the preservation of these chemicals and supplying these to developing neurons in these targets can bring about some positive things, but also negative. Several life-preserving chemicals that are supplied to developing neurons by their targets have been identified. Perhaps the most important class of these chemicals is the neurotrophins, or neurotrophins. You'll hear that in both ways. And this is what's known as the nerve growth factor, NGF. This was the first neurotrophin that was isolated per studies in 1950s and 70s. Please note that neurotrophins promote the growth and survival of neurons, and they function as axon guidance molecules and stimulate synaptogenesis. In order for us to be able to carry out smooth movement and thought process, it is important that we have cells that are specialized. And sometimes having too many is not good. Think of the success of In-N-Out when it comes down to their very simple menu. By just having 3 options and then the secret menu, they were able to generate perhaps more money than other big chains that are located throughout the world. Same thing happens with synapse arrangement. We find that neurons that fail to establish the correct connections are likely to die. And while this could be a concern for some, we find that the space that would be left by the death of the cells would be filled by sprouting axon terminals of whatever remained. This would eventually lead to very effective transmission that would improve the efficiency of whatever you and I do. So the more focused patterns of synaptic contact that we have, the smoother our behaviors and cognitions tend to be. You can see on the left side of the screen that we have a more diffuse pattern that is seen more commonly in earliest stages of development. This is when a child may not know how to walk yet and they're learning, but they're making use of their hands a lot and they're kind of wobbly and they don't have an appropriate balance. As we start developing more, and we start getting more muscle and more strength, we start realizing that our walking movement is a lot more smooth, and our thought process also should get better. And this is a result of a more focused pattern of synaptic contact that is brought about by the programming of cells that would allow some to die, but also by the environmental factors. Being stimulated with language or with other types of activities that would allow us to become more efficient at the given activity.